Hi everyone, today we're going to be talking about our work in determining the role of stiffness-mediated activity on microglia lipid homeostasis and metabolism. Just some background to get us started. Alzheimer's is a neurodegenerative disease and the most common form of dementia. It leads to the severe loss of memory and cognition. Currently, there are over 6 million Americans living with Alzheimer's disease, and that's expected to double by 2050. So we know that fat is usually associated with other diseases such as obesity, but did you know that it also accumulates in the brain? Alzheimer's disease is characterized by amyloid plaques, which are unusual clumps of the protein beta amyloid, along with other degenerating bits of the brain, including fat. To combat this, we have microglia, the resident immune cells of the central nervous system. They help with homeostasis through the clearance of fat, but prolonged activation can lead to the oxidative stress and neuronal cell death, another hallmark of Alzheimer's disease. But how do we kickstart these baddies and ensure that they're working?
With PIEZO1, of course. PIEZO1 was discovered by Nobel Prize winner Artem Panputian in 2013. It is a mechanosensitive ion channel that can be activated using a drug called UROP. Recent evidence shows that it plays an important role in microglia functions. However, what is the relationship between PIEZO1, microglia, and Alzheimer's?
Our project aims to evaluate how PIEZO1 affects microglial function and lipid metabolism. And to do this, in our experiments, we are using BV2 cells, which are microglia from mice. And we created a piezo knockdown line, and we're also using a wild type line. And we treated the cells with LPS, or we left them untreated, and LPS is used to induce an inflammatory response from the cells. And then we use different concentrations of UROP, which is a PIEZO1 agonist, and we are also mimicking the extracellular matrix stiffnesses using 1 and 280 kPa polyacrylamide gels. And from there, we are using these cells to carry out qPCR, Western blots, TIRF imaging, and flow cytometry.
These are some of our TIRF images. The flickering shows the activation of PIEZO1 channels once calcium ions flow through. This is interesting because now we can visualize changes in PIEZO1 expression and localization on the cell surface. We hope that our findings will lead to potential therapeutic targets and for more research in Alzheimer's. Thanks for listening, and make sure to keep an eye out for our next presentation to find out more.